Hi, and welcome to this short video on transcribing and translating a gene. We've talked about the processes of transcription and translation already, and you've read about them in your textbook. But to fully understand them, I think it helps to actually run through how you carry out transcription and translation to help solidify that understanding. As a part of this video, you'll do a short exercise, and you'll need the genetic code to be able to do that. So if you don't have a copy of that, as it indicated in the instructions, just pause the video, go and get one, and then restart the video because I'm going to assume that you have that. Before I move to the next slide, I want you to just think to yourself about what transcription and translation are. So pause the video, write down a quick definition of transcription and translation, and then restart, and we'll look at those processes. So this diagram shows both transcription and translation and where they occur in a eukaryotic cell. Transcription occurs in the nucleus of the cell, and it's the process of copying part of the DNA of the cell, a small section of one chromosome, a single gene, from DNA into messenger RNA. That messenger RNA is then modified, though we won't talk about that process in this video. It leaves the nucleus through a nuclear pore and links up with a two-part ribosome in order to build the protein. The protein is built by reading the information in the messenger RNA that indicates which amino acids should be linked together to build the protein. So we're going to look at both of these steps and how they occur. We'll start with transcription, how you copy DNA into messenger RNA. If you'll recall, DNA is double-stranded, and the two strands are held together by hydrogen bonds. The way they're held together is that A's, adenine, pair with T, thymine, and G, guanine, pairs with C, cytosine. These are called the base pairing rules. And so the nice thing about having these rules is that if you have a single strand of DNA, you can build the complementary strand using these base pairing rules. And that's what we'll do in this next step. So here you have a short strand of DNA nucleotides, and what I'd like you to do is put together the complementary DNA using those base pairing rules. So to start you off, C would pair with G, A would pair with T. Now what I'd like you to do is pause the video with your pen and pencil, write out the rest of the complementary DNA bases, and then turn the video back on, and we'll see if you got them correct or not. G, T, A, T, G, G, C, C, A, T, G, 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 T, G, A, G. So you can look at your paper, make sure you got them all correct. If you didn't, go back and re-look and see where you made a mistake and see if you can figure out why. But it's a fairly straightforward process, but practice helps you prevent those mistakes. Okay. The next thing we're going to want to do is take DNA and make complementary RNA. And there's one major difference when we do that. RNA does not have the nucleotide thymine. Instead, it has the nucleotide uracil indicated by a U. So when we're pairing RNA bases with DNA bases, which is what occurs during transcription, when you have an A in the DNA, it pairs with a U in RNA instead of a T. The other pairings are all the same. A DNA T pairs with an RNA a G with a C, and a C with a G. So our next step is to take a strand of DNA called the template strand, because it's the strand on which the messenger RNA is built, and figure out the complementary RNA nucleotides for this DNA. So this is the process of transcription, of building that messenger RNA. So C will pair with G, and A will pair with U, not T, because messenger RNA does not have a T. So again, like we did before, pause the video and fill this in so that you can check to see that you can do it, because this is something where practicing helps make a big difference in your competency to carry it out. All right, so now I'll read off, give the bases so you can look at your paper starting at the first one. So C pairs with G, U, A, U, G, G, C, C, A, U, G, 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 U, G, A, C. G, sorry, last one was G. So again, check these against your messenger RNA and make sure that you got the correct nucleotides in there. The major difference, again, is that anytime you have an A in the DNA, it pairs with U, not with T. Once we have our messenger RNA, we need to be able to read the information in that messenger RNA. And there are two important points to understand in being able to do that. One is that the code itself for how amino acids are indicated is a triplet code, meaning that each amino acid is indicated by a group of 3 nucleotides. And that group of 3 is called a codon because it's the part that codes for an amino acid. The second important part is that you'll notice that we're not underlining our first codon here at the beginning of our strand, but instead a little further in. And the reason for that is that all messenger RNA has some extra DNA at the beginning and the end that is not part of the gene that needs to be translated into a protein. And so the messenger RNA has a system in place so that the process of translation always begins in the correct location. And that system is to have what's called a start codon, which is always AUG, that indicates that this is the correct location to start reading this messenger RNA. And once you have the start codon, each group of 3 after that represents a codon, and I've just underlined those codons to emphasize that. Eventually, as you go down your messenger RNA, and actual messenger RNAs are much longer than this, you'll need a stopping point. And the stopping point is indicated by one of 3 stop codons, and we'll look at those in the next image. So here's your genetic code. Your book has a similar one to this, a circular genetic code. The one I gave you to print out has 2 versions. It has this one and then a tabular version, and you're welcome to use either one of those. Some people prefer one, some people prefer the other. In general, this circular genetic code is faster to use, but a little more error-prone if you're not comfortable with it, whereas the tabular genetic code takes a little more time, but you're less likely to make an error. So I'm going to teach you on the one that's a little more challenging, but again, you're welcome to use the other version if you choose. So we said earlier that AUG was our start codon. And if we were going to use this version of the genetic code to see what that indicated, the first step is always to start in the very center circle, which only has 4 choices. And those 4 choices represent the choices for your first letter of your codon. So here's our start codon, our first letter is an A. So we start at A, and then we're going to move outward to find U and then G. So AUG codes for methionine. It's also the start codon. Some versions of the genetic code will say that, others will not. To make that clear, I put it here. And on the one you have, I've also indicated it by writing it in. So we're going to use this genetic code in order to figure out the meaning of each of these codons in terms of amino acids, and then use those amino acids to string them together to build the protein. So our first codon, AUG, gives us methionine. What I'd like you to do is to figure out what each of these other codons codes for. So pause the video and then take your time, use this genetic code or the one that you have, and figure out what each of these codons code for. Then start the video again, and we'll run through the process of looking at those and making sure that you got them correct. So here is our genetic code again. Our first codon was AUG, which was the start codon and gave us methionine or MET. Our second codon, GCC, gives us alanine. Our third, AAU, gives us asparagine, which is abbreviated ASN. Our fourth, GGG, gives us glycine, GLY. And then our last codon, UGA, is one of the 3 stop codons in the genetic code. And so that tells us that this is the end of our particular amino acid, and we will stop reading off codons at that point. You'll note that the other 2 stop codons are UAG and UAA. So at this point, you've successfully transcribed the DNA into RNA and then used that RNA to figure out the correct amino acids for this particular protein. You'll have a homework assignment that asks you to do this same thing, and so hopefully you can refer back to this video if you have any trouble with this assignment. You're also welcome to contact me, and I can try to help you further. Good luck.